Number 43, what's the main purpose of the second paragraph? So we go to 21 to 31. So the previous paragraph introduces this idea of contamination of ancient DNA. And there are rules or methods by which you can try to, as they say in line 16, minimize the risk of contamination. So what does the next paragraph do? It says, in essence, however, most of these rules represent symptomatic treatment. If a sample is contaminated with modern DNA before entering the laboratory, for example, during excavation, it can be difficult to remove the contamination, and it can be impossible to distinguish between authentic ancient DNA and contaminants. The latter problem is particularly pertinent if the target DNA and contaminant DNA are from the same species, et cetera, et cetera. So even though in the previous paragraph they say that there are methods and rules that can try to minimize contamination, there are still problems that are not solved, that this is not sufficient to fix the problem. When they say it's symptomatic treatment, in other words, you are addressing the symptoms of contamination, not the cause. And so the problem is not fully solved. And so when I go to my choices, I like choice B. Assert that a solution is presented in the preceding paragraph is inadequate for addressing the full scope of a problem. Exactly. Insufficient. Explain in greater detail the nature of newly established procedures. No, it's not elaborating on the previous paragraph in that way. Emphasize the necessity of using different research technologies depending on the species. Doesn't mention that specifically. Describe the different kinds of contaminants. It doesn't do that either. So the one that is done is choice B. 44, 45. What is the author's attitude towards the technology available for identifying and removing DNA contaminants? And so we see this in the third paragraph. In the era of high-throughput sequencing, statistical tools based on DNA damage signatures are now available to assess the overall authenticity. So here's the tack. What does the author say about it? Considerable time and resources, however, are expended before contamination can be identified. And if a sample has been contaminated decades or centuries ago, blah, blah, blah. So the point is, it's expensive, it takes time and resources, and it doesn't work perfectly. It only works in certain cases. So not super, it's fine, but it's not going to solve the problem. So I think C and D are way too positive. B is way too negative. Like, it does do something. It does help. It's just not a cure-all. And we look at A, somewhat cautious, pretty mixed there, right? It's a little positive, a little negative, because this technology remains costly to use. We saw that in those lines. And indeed doesn't guarantee accurate assessment of DNA, that there's issues that are not fully addressed. So I do, therefore, like for 44, choice A. And the lines that we used were the ones that we had just read. That's 35 to 40. So 45 is D. 46, line 41, strict. Although blank laboratory exercises are often combined with bioinformatic analyses to respectively remove and identify DNA contamination, etc. So strict, hard not to use the word that's going to end up being the answer here, but really say something like particular and exacting, something like that. Right? There's very rigorous, as the answer is going to be, rules that are being followed here. And so, yeah, when I look at my choices, I like rigorous. Absolute is both too strong and imprecise. What does it mean for there to be absolute laboratory exercises? Rigorous laboratory exercises make sense. Grim, too negative. Narrow is, well, too narrow. So 46, therefore, is going to be choice D. 47, 48. What can passage 2 imply, or what can we infer from passage 2 about DNA shredded by enzymes? And we're looking from 56 to 67, so pretty narrow. And hey, what do I see right at the beginning? All healthy cells have enzymes that shred DNA to recycle it. And unfortunately, those enzymes keep right on shredding after death. Blah, blah, blah. So when I go to my choices, and I look at C, used as material to support living functions in healthy cells. Look at what it says, again, in those lines. All healthy cells have enzymes that shred DNA to recycle it. In other words, to reuse it. So yes, this is true. And the lines that we just used, 56 to 58, support that. Now, looking at the other choices, Most beneficial to scientists when the degree of shredding is high. Just the opposite. So A, B, C, D. A is the opposite. The shredding is really bad. The shredding is just what makes it really hard to follow along what's going on. That makes their scientist's job harder. Looking at B, protected from further destruction after cells die, also false. It says it continues after death. So again, B is also opposite. D, DNA is preserved by water and oxygen in healthy cells. No, again, it says specifically the opposite, that water and oxygen degrade it further. So the only one that's not the opposite of what the passage says is C. So 47 is C. And then the lines we use for 48 would be choice A. 49, what's going on with this line? 75 to 76, what scientists believe. The oldest recovered genome so far came from a horse's leg bone buried in Canada's Yukon Territory for 700,000 years. With the right sample and even better software to analyze it, scientists think they can push that back to 1 million years. So basically, the oldest recovered genome is from 700,000. They think they can push that time back to 1 million years old to recover it with this technology, etc. So a certain limitation might become more pronounced, just the opposite. Like this limitation of how long ago you can go back and get a genome will actually be pushed on, expanded. A particular boundary might be extended. Yes, the boundary of time where you can recover a genome will be pushed back or extended. It's not going to refute a theory. It's not going to restructure a chronological sequence. Don't really know what that's talking about. So 49 is B. 50. Regarding scientific research involving aDNA, both passages imply that. So what can be gleaned from both passages? Even though I think A is a bit more pessimistic, or passage 1's a bit more pessimistic than 2, which is kind of laying out some of the new techniques and technologies that fix the contamination problem. What do they have in common? And what they have in common is C. Evaluating traces of aDNA is generally problematic. And they both agree on this. Like passage 1 is saying, there's so much contamination, you can do a little bit to fix it, but it's something that's a really hard problem to solve. B agrees that there's contamination, that there's issues, there's this shredding that happens, right? Contamination, corruption, shredding, so all these problems. But the difference is that 2 is kind of focusing on the breakthroughs that have made this not as bad of a problem. But both of them agree that it is a problem. And so 50 is going to be C. They don't talk about, for choice A, smallest quantities containing trace of contamination. If anything, when you shred it, there is no contamination, but it's shredded, so you're losing all that information. The analysis of aDNA has led to a greater understanding of RNA. Doesn't talk about that specifically in both passages. D is probably not something that's supported really by either. I think they agree that aDNA analysis is useful. It's just hard to do. So 50 is C. Given the discussion of the RNA bait technique in the last paragraph 2, would applying that technique be useful in the circumstances in those lines? So the last paragraph, they talk about this RNA bait thing. And it says, if searching for human DNA, researchers would prepare the bait by preparing millions of strips of human RNA, blah, blah, blah. When mixed into the sample, this RNA gloms onto the human DNA and only the human DNA. So the idea here is you can use these little RNA baits to pull out DNA from particular species. If, for example, you're looking at the DNA of some horse and you want to remove human DNA, you can use these RNA strips to do that. So would this work in what's described in 27 to 31? So here they're talking about contamination from the same species. And so here's the problem. This trap only works if you can remove human RNA, for example, from some other species DNA, right? If you can only remove human DNA from a horse's DNA or something else. If you're working with human DNA and you have other human DNA that's contaminating it, this RNA bait trap thing is not going to work. So therefore, would this be useful? The answer is no, because as we see in A, the RNA bait technique is used to isolate DNA of one species from contaminant DNA of other. So 51 is A. 52, what's the relationship between the two passages? So they have some agreement. I would say it's more of a mixed relationship. They agree on the problem. I think 2 is a bit more optimistic in what can be done. And So I look at B. Both passages examine the difficulties in a line of research, but passage 2 gives more detail about how they're trying to address those difficulties. So yeah, B is laying out, here are some actual ways, techniques, technologies that can be used to fix this problem. 1 doesn't really do that. It focuses more on the problem. I wouldn't say that both passages consider a proposed approach for solving a problem. Passage 1 is really just laying out the problem. Both passages present hypotheses that are untested. We don't see hypotheses being discussed here. It's just talking about technology and what can be done and what can't be done. And I don't think passages are raising concerns about the validity of research findings. And I don't think 2 effectively articulates the logic underlying the concerns. This is just not what's mentioned. It's talking about a problem and how that problem might be solved, and 2 goes into more detail about some breakthroughs that could solve that problem. And so B captures what's going on in those passages.